Okay.
Yeah, Diane, this is Steve. Thanks for the invitation. And Rick is the one who's kind of taken the lead on all of this. I joined the game relatively recently. He's been at it for a number of years. So I will let him take the ball on this.
And Julie from the Garden of the Salish Sea is here also. I don't know if any of your other cohorts are with you or not. So
I think some may join or maybe not. I'm not sure. But if I can, I'd like to share my screen and give me a thumbs up if you see my screen.
all right.
And can hear me
Yeah,
okay.
you're good.
Okay. That all working?
Yep.
Okay. Well,
Yes.
yeah, thanks. Thanks for inviting us.
You
Speak.
are in, sorry, we're seeing your like presentation mode. So if you have notes, we can see your notes and all of that.
I don't have notes. This is all off the top of my head, Eleanor, as you know.
Yeah. Great.
But yeah, good to see a couple familiar faces, by the way. I'm Rick Beauregard, past chairman of the Drayton Harbor Shellfish Protection District Advisory Committee. And with me today is one of our team, Steve Wilson, who joined us this summer. And maybe coming on board will be a couple members of our team. And Julie, our partner from Garden of the Salish Sea. And we're here to talk to you about our the Drayton Harbor HAB Hunters, Harmful Algae Bloom Monitoring in Drayton Harbor. We got a great little group, citizen group of people, and in collaboration with the Garden of the Salish Sea, we've got a couple student interns that are involved. But this is a program we started in summer of 2020. It's kind of an outgrowth of my involvement in the Shellfish Protection Advisory District. You know, that organization is very focused on water quality, in particular, you know, fecal coliform, and you know, there are many other dangers to our shellfish resources, and one of them is harmful algae, and I had some background on that, and so I looked into whether we were doing anything here and you know, what we might do, and I learned that we do have an organization called Sound Toxins, which we have also collaborated with, and the Puget Sound Restoration Fund is doing some things. We have a dual purpose, and that is sentinel monitoring for harmful algae, and then the education component, which with Julie's help, we're focused on. And we are self-funded. We've had some donors to help us kick off and get some equipment and get us started a couple of years ago. So Sound Toxins is a network of citizen scientists, organizations like us and others that are a little bit more organized than us, monitoring for harmful algae up and down Puget Sound and the coast of Washington. Oregon has a similar program. California, Alaska, and British Columbia also have similar programs. So what we are doing is we're sampling for harmful algae, HABs. It's easier to say HABs, so I'm going to say that from here on out, HABs and other phytoplankton. And the other phytoplankton part is important, I think, and we'll talk a little bit about that later. So we sample weekly during the spring and summer through fall, and then biweekly during the winter months. We're doing microscopic analysis on plankton net tow and counting relative abundance and quantitative abundance in whole water and concentrated samples, and collecting some other meteorological and hydrological data, and reporting our data to Sound Toxins. What are phytoplankton? Steve, maybe you can jump in here and help. We're going to just give a little quick primer for those of you who need a little primer, and I don't think many of you do, but we'll go ahead anyway quickly and talk about what are phytoplankton and why are we monitoring them?
Yeah, phytoplankton are they're all single-celled microscopic, tend to be microscopic organisms that use photosynthesis to convert sunlight to food. And in the process, they release a whole lot of oxygen and take up a whole lot of CO2. And they are at the bottom of the food chain and probably the single most important organism to every other life form in the ocean, freshwater and saltwater. But we broadly, there are two groups. The diatoms are they actually have a silica glass shell that they form, and they're absolutely exquisite. You'll love seeing them. They're beautiful little organisms. And the dinoflagellates. And the dinoflagellates are basically a cellulose, effectively a paper. If they make a shell, it's a little paper shell, and they have the a couple, depending on the type, flagella that they use to move around in the water column with. And we're primarily concerned with the organisms that produce toxins. So every time we do our screening, we're looking for specific organisms, but we're also looking at the overall kind of makeup of the diatoms and the dinoflagellates in the water. And we can, Rick has done a lot of work on kind of predicting trends based on what we see happening with the dinoflagellates or the diatoms at any given time. And we're hoping that over time, as we continue to build up our database, we'll be able to be even more predictive about when we can expect to see, for example, the harmful algal blooms happening.
So while we were out there one day, we had some folks from the Swinomish Crab Abundance Monitoring Program come out. They wanted to deploy their trap on our pier, and they asked us if we would help them as well. So our intrepid little team volunteered to monitor and pull their trap every week. And so we did that this summer. And then in addition, I actually approached the Department of Health about doing their biotoxin monitoring, mainly because I wanted to get access to their data on a regular basis so we could compare our phytoplankton and HAB data with the actual biotoxin data that's coming from the mussel sampling that they do. So they do a sentinel mussel sampling all through Puget Sound. We've been involved in this just since August, and we're sampling at Birch Bay, Village Marina, and Semiahmoo Marina, doing weekly sampling during the potential bloom months, and then sending our samples off to the lab at the Washington Department of Health lab for analysis. So why are we doing this? Well, I mentioned, I'm involved in the Shellfish Committee, and we're focused on fecal coliform, but you might argue that an even bigger effect on our shellfish resources is harmful algae. And so I'm sure you're all familiar with these signs in 8 different languages, I think. We're doing it to really provide a kind of a potentially early warning of harmful algae, collect some information that the Sound Toxins Group, which is a group housed in University of UW in the NOAA Sea Grant program that sort of coordinate the Sound Toxins Group, but to help provide some information and see if there's a way we can predict when the blooms might come and how bad they might get, and that sort of thing. The state has limited resources for this, and this is why they've asked us to do the sampling for them. They have to drive throughout Puget Sound to collect these samples, and so they rely on citizen scientists like us to do the sampling and the phytoplankton sampling as well. We're doing it to protect recreational, tribal, and commercial shellfish harvesting and economic impacts. It doesn't do anybody any good if we're closing the resource when we don't really need to, or we're not closing it when we should be. And that's the bottom line, is to protect public health. We're also doing it because Birch Bay Village Marina, for example, had the highest paralytic shellfish poisoning toxin counts concentration of record in Whatcom County between 2010 and 2022 this year. 7,500 micrograms per 100 grams of tissue in mussels, and that was in June of 2010. Drayton Harbor had the second highest, nearly 7,000 in that same season. Half of the top 10 highest PSP results since 2010 in Whatcom County were recorded in Birch Bay and Semiahmoo, Drayton Harbor. And Whatcom County, looking at entire Puget Sound, had some of the highest PSP values on record. We're getting closures that are happening earlier in the spring. Used to be in July, now it's coming in May, and they're going later in the fall. This year, in 2022, we had a closure that went well into October. It was just lifted last week. And as Tracy Barry from Department of Health has said, every time we want to reopen, a new bloom will show up. And so, you know, they've been calling us lately to confirm that it's safe and that, you know, to see if there's any organisms in the water, to see if they could be thinking about at least opening the resource. Another reason why we're doing it, this picture on the left, these are the sound toxin sites that are being monitored on a regular basis. And then I didn't put the British Columbia sites, but there's some up there. But as you can see, you know, we don't have any sites here in our area. We do have a commercial shellfish operation in Drayton Harbor, as you probably all are aware. So that's the left-handed picture. We don't have coverage up here for sound toxins, so we thought we needed that. There's occasional sampling, or there was sampling in Birch Bay apparently, but I don't know if that's a regular thing. The other reason really is depicted in the picture on the right, and that shows the influence in our area of Fraser River, and you know, how it has a huge influence, particularly on our area, that will influence the phytoplankton and the growth of harmful algae. But yeah, the Fraser River contributes the vast majority of freshwater input and nutrients and pollutants and everything else that goes with it to the entire Puget Sound area. If you added up all the other freshwater sources, Fraser River would dwarf all of that. But yet we didn't have any sampling sites up here. So I felt that this was a strategic location. We needed to fill that gap in and provide that data for full coverage of our resource. So where are we doing it? Here are our 3 stations that we're doing currently. We have 2 years of, 2.5 years actually, data at Drayton Harbor. We've been sampling here at the Plover Dock at the end of Semiahmoo Spit. And then this summer, we've started sampling in the marina, in Semiahmoo Marina, and at Birch Bay Village in coordination with the mussel sampling that we're also doing. So now we'll have, you know, data, real-time data that will coordinate with the mussel data, and I'll show a little bit of information about that. We have very limited to show you so far, but we do have some. So what we're doing, we're going out and getting weekly samples and data, doing the microscopic identification and quantification of all the phytoplankton species present, at least down to the genus level in the net and the whole water samples. And we're collecting the live mussels that we send to DOH for laboratory analysis at Shoreline for paralytic shellfish, diuretic shellfish poisoning, and amnesic shellfish poisoning. So that's Alexandrium, Dinophysis, and Pseudonychia. So what are we finding? This year, 2022, has been a PSP year. We've had quite a bit beginning early. We've had HAB species present pretty much throughout the year. One of the 4 target species we're looking at, in addition to the 3 I mentioned, we also are watching for heterosigma, which is more of a concern for fish farms than it is for shellfish. And we don't have any fish farms. So our main concern is Alexandrium and Dinophysis. Pseudonychia is a concern, but it's more of a concern out on the coast, I think, at least right now. It's spreading, but we're watching for all of them. And we have them throughout the year. Alexandrium was present in 16 of our weekly samples between April and October. We had PSP, paralytic shellfish poisoning toxin, above 80 micrograms, which is the safe standard. It's a national standard for 7 weeks, starting in June in the mussels, in the mussel tissue. And we've had 2 Department of Health recreational shellfish closures for PSP this year, first one starting in June, and we went through mid-August and they lifted it, and we didn't see any cells in the water, and then we started seeing them again, and then we started seeing the toxin in the mussels again, and so they closed again in October 6th, and that was just lifted last week. So those are a couple of the things, and here's the announcement that came out. This is a picture of Alexandrium concentration at Drayton Harbor, Plover Dock, versus PSP concentration, the toxin in the mussels at Drayton Harbor, Semiahmoo. So they're different stations, but we don't have data of the plankton at Semiahmoo, so it was the closest we could come to, and this is for 2022. But you can see there is a correlation. The numbers that we count, the blue is the Alexandrium cells per liter that we're counting in our plankton samples. And when they started to increase back here in May, then we started to get an increase in the PSP toxin in the mussels. And you can see we had a big peak here, and then the concentration of the mussels peak. So there is definitely a predictive capacity to determine when we're likely to have concentrations of PSP in the mussels. It's not a one-to-one correlation, and it's hard to know how bad it's going to be. And I'll talk a little bit more about that later, but this is kind of what we're trying to accomplish, is to try to figure out what's coming. Why is this happening, and why is it any different than any other year? Well, that takes a lot more data, but here I've just plotted some precipitation and water temperature data for Drayton Harbor, Plover Dock, from 22 on the top and 21 on the bottom. And you can see they're a little bit out of sync. You know, we had a lot of rain last year, and rain in the spring went all the way through June until it stopped completely. Whereas the year before, we had that period, if you remember, in June, where it was super hot, and that heated up the water pretty quickly. And we had a completely different regime at that time. So, you know, it's hard to draw conclusions from any of that, but that's what we're trying to monitor. One thing we did notice, though, this year was when we had some pretty high blooms of diatoms in the early spring, and they suddenly crashed, like within a week. All of a sudden they're gone, and what was replacing them was more dinoflagellates and other flagellate type forms. And so I looked at the ratio of diatoms to HABs, or I'm sorry, diatoms to total numbers of plankton. So what's the percentage of the total number that is diatoms? And that's this blue bar. And you can see, you know, we're 80 or 90% diatoms for most of the year. But then here in June, that number crashed, and we went down to below, you know, about 15% were diatoms, and the rest were other forms. This other line, this gold line, is Alexandrium concentrations. So you can see that, you know, when the diatoms, and we're not quite sure why this is happening, maybe they're consuming all the nutrients or consuming one of the nutrients or whatever, but they're creating a niche. And what's coming in to fill it are the dinoflagellates and flagellates, and specifically some harmful algae among them. So my question here, and we're going to have to take some more data and look at some more results, but, you know, when you're trying to find something, maybe you don't look at the thing you're trying to find, you look at something else. And so maybe what we really ought to be doing is looking at the diatom populations and their bloom characteristics to see if that will predict what will be happening with the dinoflagellates. So that was interesting information that we got this year. What are some of the causes of these HABs? You know, there's a multitude of things that it could be, but certainly rain and runoff and nutrient loading. We had huge floods last year, so we know that, you know, we got a huge pulse of nutrients coming into the water bodies. We had less solar radiation this spring, and we had a lot of mixing from winds, and the mixing churned up spores from the bottom sediments. So these dinoflagellates, Alexandrium in particular, form resting spores, and they settle to the bottom, and they sit in the sediment until there's a time that's more conducive for them to grow. So as summer approaches, they get pulled up into the euphotic zone again, and they start blooming. And so we think that's probably one thing that could be happening. And then summer comes, we have a little bit more conducive situation for bloom conditions. And one might be the crash of the diatom population, and there's a niche there for them to fill. A couple other things that we've kind of observed or learned. Obviously, HABs are commonly referred to as red tides, but not all red tides are red. This is a picture of taken by Mark Seymour at Drayton Harbor Oyster of a green tide. This is a bloom of Paramimonas, which is a chlorophyce flagellate. It's not a toxic bloom, but when you have a bloom like this and it starts to die off, then it can consume oxygen and can be a fish kill problem and cause other water quality issues. So it can be a noxious kind of a problem, if not toxic. But yeah, not all red tides are red. Harmful algae blooms are not all blooms. I mean, one of the things I think that strikes me the most from doing this for the last couple of years is how low a concentration of cells in the water column it takes to create an impact in the toxicity of the mussels. We have very relatively low concentrations. I can't say we've seen a bloom of Alexandrium in terms of numbers of cells per liter, and yet we have relatively high, well, I won't say high, but over standard health standard levels in the mussels. And so that's concerning in terms of, maybe we need to find a better way of predicting them. Not finding it doesn't mean it isn't there. There's a tremendous amount of three-dimensional spatial variability in these populations. We sample surface and we sample a column from bottom to top. And if there's a lens in there of a bloom that's occurring, we might catch it. If not, we won't. We'll just catch a few that are floating around in the water column. But they may be blooming like this green bloom somewhere in the environment, and we're not catching it at our particular sample site. So that's another reason why we need to be conservative in terms of closures and looking at these data. There's also a lot more temporal variability than I had expected. This bloom here on the right, this green bloom, within a couple of days, it was non-existent. I mean, the cells were still in the water, but you didn't see a green bloom like this. And as I said, this last bullet doesn't take high concentrations to show up in the biotoxin results. I wanted to touch on shellfish safety. We went to the Oyster Fest in Blaine last week, and I got a lot of questions from people about, gee, are the oysters safe? Why are we having an Oyster Fest, but we have a closure? And so I think this is important maybe for you all to spread the word, I think, because I think the public has a misconception about this. But so there's several different types of things that may affect shellfish safety, and not limited to the ones on this page, but we can divide it into two domains, really: recreational and subsistence, and commercial. And we have the harmful algae, we have fecal coliform and general water quality, and then we have something called Vibrio, which is a bacteria that occurs in very warm water, and it does affect us, in the heat of the summer when tides are low and clams and oysters are sitting there in that low tide and the heat comes up. You can't have a Vibrio outbreak. So the way that these are dealt with, all of these three and these two domains benefit from the sampling and the monitoring and the closures that we do. But on the commercial side, they actually sample the product, and that's a law that's required by law. for HABs and for Vibrio too, when it's Vibrio season. So, you know, if you're buying it in a store, or you're buying it in a restaurant, you know, it's probably safe. However, if you're not, and you're out there, you're just picking clams, then you know, you can't rely on it necessarily being safe. And so before you dig, you need to check out the shellfish safety information maps that are published and updated daily by Department of Health. And so here you can see, this is 2 days ago, I think, when I downloaded this particular one, but there's still this crosshatched, red crosshatch area all around the peninsula here is biotoxin closures for all species. We still have a little bit of that up in our area, Bellingham area, but you can see North Whatcom County is open now as far as biotoxin closure is concerned. But for the recreational shellfish fisher, they also need to look at the beach closures for other reasons. So if you zoomed in on the previous map to Drayton Harbor and Birch Bay, this is what you'll see. And the top one here are commercial growing areas. These are the splotches, and the recreational shellfish beaches, these are the lines along the beach. And both of these are depicted on these maps. So you need to look at these maps if you're planning to go out recreational shellfishing and make sure that your beach is safe. We have in Drayton Harbor, we have the approved area, and then this conditional area. It's conditional on rainfall primarily. So certain times of the year, or when a certain level of rainfall happens, those conditional areas will be closed, and that's primarily due to fecal coliform. And then there's the areas that are closed due to pollution, and again, mostly fecal coliform. This is due to the inputs from Dakota and California Creek, and the outfalls and stormwater off of Blaine, and the marinas, and the outfall from the sewage treatment plant. So yeah, I think the message is basically if you're going to a restaurant, it's probably safe because they test, even though the beach out in front may be closed. But if you're going to go dig for recreation, then you need to check this map. So hopefully you can help spread that word. People have questions. What's next for our DH3, Drayton Harbor HAB Hunters? Well, now we have to call ourselves the HABs, crabs, and mussels hunters. So we'll continue with this, the HAB monitoring and reporting and submitting our data to sound toxins. We're hopeful because of COVID, they weren't recruiting any new sites. They were glad to take your data, but we weren't really part of the program per se. And we're hopeful we can become part of the program because they've provided some resources to do some additional sampling congruent with what we're doing for nutrients and chlorophyll. And we'd really like to get some nutrient data to start making some conclusions about our sites. We'll continue the Sentinel monitoring program for, as we're a representative of Pacific Shellfish Restoration Fund in doing that for Washington Health Department. And I don't know if our team is up for it. We might even add some sites to that if they're interested. I know they have a hard time getting up into Point Roberts on a regular basis. And then the Swinomish Crab Abundance Monitoring Program, the SCAMP program. Hopefully they'll ask us to do that again next summer, because that was great fun, and it was great for our interns. We have 2 interns working with the team from Blaine, and yeah, that was a real good hands-on experiment and project that they really enjoyed. And then I continue to be involved with the Drayton Harbor Shellfish Protection District, and Whatcom County is doing the fecal coliform, and the state's doing the marine sampling. So I'm involved in that. And then again, more opportunities for some of our student interns, which would be great. So at that point, questions and answers, if we have time. And while you're thinking of questions and answers, I've got a few photographs that Steve can be discussing, and jot down your questions, put them in the chat, or say them out loud, and we'll see what we can answer.
All right, first picture is just Alexandria, the one we've mentioned a number of times, that causes the paralytic shellfish poisoning. And you know, as a new member of the team, I had difficult time trying to figure out exactly what these were until I could find them in a string like this, because when they're individual cells, it's hard to differentiate them from other things. But if you're lucky, they form up in strings like this, and then you know exactly what you're looking at. But they are a dinoflagellate, and you can see a little bit of the flagellum potentially off them. But yeah, go ahead and step forward here. And then these have a very characteristic shape. These are easy to identify as the dinophyces, but then there's 4 or 5 different subgroups that make it somewhat difficult to figure out exactly which one you're looking at. But again, just they're easy to spot, very interesting little organisms. This is the only one of the diatoms that we are particularly interested in. This is the Pseudonychia, and they are the long, skinny diatoms that form up in chains, and I'll often see these 20, 30, 40, 50 cells long in long chains, and motile. They'll be moving, and it's really interesting to figure out, from my perspective, how the heck does a chain of 50 organisms decide which way to go? But okay, and this is Nautiluca, and Nautiluca is we do get a little bioluminescence. I don't know if any of you have been to the bioluminescent over in Bellingham Bay, but it's caused by this organism, and when they're mechanically agitated, they will produce a lovely blue light. But if you've ever seen that sea sparkle at night, that's what that's coming from. Now, this is skeletonema under the light microscope. But what is really interesting about this one is if you put it under a scanning electron microscope, which I have access to at Western Washington University, they have 2 there, and I have access to them. Anyway, and you start looking at the joints between the individual cells, the kneecaps on those cell connections are just astounding to me. That's magnified something like 70, 80,000 times to be able to see those kneecaps, but incredibly delicate, beautiful structures, and again, all made of glass. And this, we have a Dungeness crab larva, and they have beautiful blue eyes as a larva. And I was surprised the first time I looked at them, but they're gorgeous little animals with these beautiful blue eyes.
These are barnacle larvae.
These are barnacle larvae, huh? Okay.
I can't help when I look at some of these pictures and the pictures coming out of the Webb telescope of thinking, I'm looking at the universe in a drop of water, and it really does strike me that way. Some of these photographs. Some sort of snail larvae, and up here on the left is a diatom called Diddledum, Brightwellii.
Oh, is this when we were doing the larval collection, and we have a light trap, and when we empty the light trap, sometimes it's full of crab larvae, sometimes it's full of these lovely polychaete worms, some of them quite impressively large.
This is one of the ones I was referring to. It looks like the monkey nebula, but it's actually a very close up of a black claw crab larvae.
Yeah, the patterns in them are gorgeous.
And for a size reference, you can see this string of Thalassia syra diatoms up by his antennae. These are a couple of things that came out of the light trap. This on the left is an isopod that's pretty common. He's a cute little booger. And then this isn't light trap on the right. It's the microscope plankton sample, but this is a polychaete worm larvae. We did have a couple questions and comments. One was, is the new floating oyster bag growing technique being used by Drayton Harbor Oyster an environmentally safe technology for Drayton Harbor in the Salish Sea? And what are the risks? Well, generally, oysters remove a lot of pollution from the water. They pump like 50 gallons of water a day through their filter feeding mechanism. And so they tend to clean up anything floating in the water. So from that standpoint, from water quality standpoint, I think generally they have a positive impact. Now, you know, in a situation where there's intense farming anywhere like this, whether it's fish or shellfish, then, you know, their waste products may also enter the fray. I think a lot of the issues with shellfish farming have to do with the materials they use, the bags, the tubes, if it's gooey ducks or whatever, and they get away, and, you know, so we have a lot of plastic pollution in the environment. So that's certainly not a good thing. And, you know, there'll be some controls on that, I'm sure. You know, it has a negative, I think, a negative impact from an aesthetic point of view. If you're looking out on Drayton Harbor, you may not care to see these floating arrays out there. It's not going to be huge, but, you know, I think to the folks along Semiahmoo, Drayton Harbor shores there, they may take issue with that. So is it environmentally safe? Yeah, I think there's pluses and minuses. I think the way they're doing it now, it has a much more potential to harm the seagrass. The way they'll be doing it in the future is that because of these floating bags, they'll be able to basically drive in on their boat and harvest the bags rather than having to go out on the beach and pick up the bags and tromping along all the whatever's there. I know they've cited it so they have a minimal impact currently on the current operation, or try to. But yeah, impact on the seagrass might be an issue. I think a positive thing generally. Maybe shade's an issue. I don't know. That would be for someone to look at. And then I don't know if that answers your question. Probably a lot of questions on that. Dinoflagellates are generally indication of poor water quality. You know, dinoflagellates have been around forever. Harmful algae blooms have been around since prehistory, and human, you know, European contact. There's some documentation of that. I don't know if I'd call it poor water quality. It's definitely a different water quality regime. I think warmer temperatures, and that kind of coincides with other things, higher nutrient levels and so on. But they exist and they're out there, they're natural. And they can take advantage of poor water quality situations. And because they're mixotrophs and not just simply primary producers, they both produce oxygen and they consume oxygen. So they do both. Some do more than others. And they consume diatoms and bacteria and other things. So they're predatory even. But you know, they're part of the food web. So it's not, it's probably not quite correct to say they're not healthy, or they're, you know, they're indicator of poor water quality. At least that would be my interpretation. Any other questions?
I've got one comment and then a couple of questions. This is Lyle Anderson. I was one of the two, I think, that commented on the new installation that's going to go, the oyster thing that's going to go in the west end, I guess, of Drayton Harbor. And I was concerned that, you know, with the overall decline in the bird populations, and that includes marine birds, that do we really need this another insult to the natural environment, especially because, you know, Semiahmoo, Drayton Harbor, both sides, Drayton Harbor and the ocean side, you know, there are a lot of birds there in the wintertime. So, you know, I'm concerned about that. So anyway, and the response I got, it didn't seem to really address that. But anyway,
Yeah.
I'm just wondering if the the fecal coliform situation, Dakota Creek, California Creek, that's been going on for years, and I've been hearing about it for years. Um, why is it still a problem? I mean, is there that
Yeah.
many sources of fecal coliform that they can't get control of it?
Well, fecal coliform are contributed by warm-blooded, and they're an indicator of human, but they don't come only from human populations. They come from cows, they come from animals, dog poop, birds, and so on. And actually, the upstream water quality, for the most part, in Dakota Creek and in the California Creek watersheds, has improved quite a bit because of efforts that have been undertaken, spearheaded by the protection district over 25 years, way before I got involved in it, to get farmers to handle their manure better and to make buffer zones around creeks and so forth. So, and then of course the replacement of the old sewage treatment plant and the sewer underneath Drayton Harbor was a huge impact. I think that was the reason why our water quality was so horrible, and why that fishery was closed for so many decades. So that's all been a positive on the water quality. It still exists. It's like the air quality in California. They have the strictest air quality requirements in the world, but when the population is growing the way it is and everybody has 2 or 4 cars in their yard, we still can't get our arms around the problem. And that's kind of what I see as the next problem here is development, land development. We're the place that's left between Vancouver and Seattle to develop, and it's going to develop, and that's going to have an impact on water quality. So we're enjoying our oysters now, but we may not be forever. And we have a challenge with the water quality, keeping it safe. And that's one of the reasons why Drayton Harbor oyster is moving to the other side of the bay, because that's had consistently good water quality, whereas on the other side, it's infringing, that little island of green water is getting smaller and smaller. And so they're hopeful that they'll be able to operate there longer. I agree with you. The industrial level aquaculture that you see in a lot of places, it has a huge impact. This is a small family farm. There aren't any other farms that are going to be here, probably. And, it's a relatively small operation compared to some of the ones that I think do have a huge impact. And the truth is, shellfish farming or harvesting has been going on here time immemorial. So I'm not sure what's going on with the farming aspect has any greater impact on bird population, for example, than just the natural environment. But
my other question was, so is it possible that when people are told not to harvest because of the different toxins and algal blooms, like for instance, people digging clams on Birch Bay, and then is it possible that the oysters that are harvested at the same time could test okay, as opposed to clams in another area? I mean, is it so local? Can it be so localized?
yeah,
And the
yes,
corollary with that is, where you're testing in the, I don't own a boat, so I don't spend a lot of time in the marinas, but the time I have been out there, they seem to be, the water doesn't it seems to be trapped and warmer than it is out, in the bay, Boundary Bay and such, Salish Bay. So, can it be localized to that extent? And could your test results be affected just by the location that you're sampling?
yeah, and I think that's the reason again why we felt that we needed samples up here, because you you could close the entire Puget Sound because you got a bad sample down in Bellingham, where up here the water's clean. And so having more sample points, and this is where the citizen science comes in, is, you know, that project is an impossible publicly funded project. It won't happen. But with citizen scientists out there pulling samples and getting more data points and providing that data, we get a much better coverage. And it's true of a lot of kinds of projects, I guess, but in this case, it definitely is. And so that's the goal, is that we're not closing when we don't have to, and we're closing when we should be. And we're keeping them closed for as long as we had to, but no longer, you know, because it does have an economic impact, as well as an impact on the community and the tribal communities and so forth. So, and at the same time, being conservative and protective of health. So the health department is always going to be way more conservative than you think they ought to be sometimes. And they're going to close a whole area when they get a sample that looks like it's hot. So unfortunately, you can't have coverage everywhere you'd like to.
Thank you
Good
for
question.
doing this, and those
Good
are
question.
awesome photos. Awesome
Yeah.
micro.
Yeah, you get a sense for why we're doing this, which I didn't cover, which is it's just fun. Just looking in that microscope and seeing those pictures is really cool.
The
So
thing that I've been most surprised by is that as we sample week to week to week, it's like each week is a brand new slate of characters, and you know, things that I just learned to identify the previous week are all gone. And now I have brand new things to try to figure out what I'm looking at. I am surprised that it changes as dramatically and as quickly as it does. If you have a microscope, come join us or get one.
yeah, we'll teach you how to recognize them. It's not that hard. Diane, I think we're out of time, but I'll leave
We
that up
are
to you.
just about, we got 8 minutes, 7 minutes. Thank you so much, Rick. That was fascinating. Steve, thank you so much. Steve, thank you for letting me know or being introduced to you, I guess, so that we can make this chain go a little further. Thank you so much.
Great,
And
thank you.
anybody else